We can now move on to our discussion on the amino acid derivatives. Many important compounds within the body are derived from amino acids. Phenylalanine and its derivative tyrosine are precursors to the catecholamines, which will include your dopamine, your norepinephrine, and epinephrine. The thyroid hormones are also derived from tyrosine, and the pigment melanin is going to be derived from dopa. Tryptophan is going to use vitamin B6 to make niacin, which is the precursor for the electron carriers that we've already talked about, NADH and NADPH. So as a quick aside, how many ATP does NADH make when it exchanges its electrons in the electron transport chain? NADH is going to exchange its electrons to make 2.5 ATP per NADH molecule. And then our FADH2, the other high energy electron carrier, is going to synthesize just 1.5 ATP. Using vitamin B6 and also the cofactor tetrahydrobiopterin, which simply means that it's going to transfer a hydroxyl group to a substrate, this tryptophan can be converted into a serotonin. Serotonin can then be in turn converted to melatonin, which is useful in sleep. This is why people associate eating turkey on Thanksgiving with being tired after a meal, since turkeys have a lot of tryptophan, which then gets converted into high levels of melatonin. Vitamin B6 is also going to be used in the conversion of histidine into histamine, which, as you know, is important in the allergic response. Now let's sidetrack just for a moment and talk about heme synthesis. A third use of vitamin B6 is in the very first step of heme synthesis, which is the conversion of a glycine into a porphyrin, and this requires vitamin B6. With that said, can you think why a vitamin B6 deficiency can lead to sideroblastic anemia? So the first step of heme synthesis is going to be the combination of glycine and the succinyl-CoA by the enzyme ALA synthase. This will make your molecule of delta aminolevulinic acid, and this is going to be your rate-limiting step of heme synthesis. This is going to require vitamin B6 in the process. So this is why a deficiency of vitamin B6, you're not going to be able to proceed with the very first step of heme synthesis, and that'll lead to your sideroblastic anemia. The amino acid arginine can be used to make creatine, urea, and nitric oxide. And then lastly, glutamate is going to be a derivative of the inhibitory neurotransmitter GABA using the cofactor B6 as well. Notice a trend. And glutamate can also be used to make glutathione, which is an antioxidant that we talked about earlier, that remember it needs NADPHs to be regenerated. So we have the same pathway that we just saw with the amino acid derivative slide. It was a pretty basic pathway, so now let's fill in the picture just a little bit. The catecholamines, which include the dopamines, norepinephrine, and epinephrine, these are the hormones that are derived from the amino acid phenylalanine. The first step in catecholamine synthesis is the conversion of phenylalanine into a tyrosine via the enzyme phenylalanine hydroxylase. So here I'm just going to take this step of our pathway and just blow it up a little bit so we can see it better. This step is going to require tetrahydrobiopterin as a cofactor, which after the reaction proceeds, they'll get reduced into a dihydrobiopterin. So we're going to need a tetrahydrobiopterin for this reaction to proceed. Dihydrobiopterin reductase is used along with an NADPH molecule to restore our dihydrobiopterin into our tetrahydrobiopterin so it can be used again. Speaking of NADPH, what disease results in a decrease in NADPH synthesis, which can ultimately lead to red blood cell destruction and bite cells? Uh-oh, I said a buzzword, bite cells. This highly tested, high-yield, all-important disease that I'm going to be alluding to is G6PD deficiency, where the enzyme glucose-6-phosphate dehydrogenase is going to be missing or defective, thus leading to your bite cells. The second step of our catecholamine synthesis is going to be converting our tyrosine into a molecule of dihydroxyphenylalanine, or DOPA for short. This step is going to require the enzyme tyrosine hydroxylase, and again it's going to use tetrahydrobiopterin as a cofactor. This step is very similar to the one that we just saw, just with different enzymes which are very well named. This one is tyrosine hydroxylase, while our other one was phenylalanine hydroxylase. Same step otherwise. In the third step, DOPA is going to be converted into dopamine using vitamin B6 and also DOPA decarboxylase enzyme. Now this is the step in catecholamine synthesis that's going to be inhibited by carbidopa, which is going to be used to treat Parkinson's disease. As you'll see in the neurology section, Parkinson's disease is caused by having too little dopamine in the brain. So that leads to the question, why do you think a drug that blocks dopamine synthesis actually helps patients with Parkinson's disease, meaning they have too little dopamine in the brain? Well, that's because carbidopa only works outside of the blood-brain barrier in the periphery. So carbidopa only works in the periphery. This allows more dopa to be available in the brain for the conversion into dopamine, and then this way there will be a higher level of dopamine in the brain. So let's get back to our catecholamine synthesis pathway. Dopamine is then converted into norepinephrine by the enzyme dopamine beta hydroxylase, which requires vitamin C as a cofactor. And now that I'm thinking about it, do you remember what pirate collagen disease results from a deficiency of vitamin C? Well, yeah, it's scurvy, which we talked about earlier. Do you remember how scurvy presents? Scurvy is going to present with impaired wound healing, swollen gums and bruising, corkscrew hair, perifollicular and subperiosteal hemorrhages, and also hemarthroses. This is my favorite pirate disease because pirates would lack vitamin C while out on the boat. What's a pirate's favorite letter? Well, you may think it's R, but his true love be the C. So just remember, vitamin C is also used in the catecholamine synthesis pathway, where here we see it as a cofactor to help convert dopamine into norepinephrine with the help of our enzyme dopamine beta hydroxylase. In the final step, our norepinephrine is going to be converted into epinephrine by the enzyme phenylethanolamine N-methyltransferase. Since this reaction is going to add a methyl group to our norepinephrine to make our epinephrine, it shouldn't be surprising that SAM, the methyl donor man, is used as a cofactor to donate that methyl group. Now this enzyme is going to be regulated by cortisol. So stress is going to increase cortisol levels, and cortisol is going to activate this enzyme. So when cortisol levels increase due to our stress, more epinephrine is going to be produced. You may also want to keep in mind that both dopamine and norepinephrine are produced in the central nervous system and in the periphery. However, epinephrine, on the other hand, is produced only in the medulla of the adrenal glands. Now that we've spent all of this time and energy just to build up some catecholamines, let's go ahead and tear them apart. How do we break these down? Catecholamines can be broken down by our Pac-Man enzymes, monoamine oxidase, or MAO for short, or catechol O-methyltransferase enzymes, which are COMT for short. So dopamine is broken down by our MAO or COMT into homovanillic acid. Norepinephrine is broken down into VMA or vanillyl mandelic acid, and lastly, epinephrine is broken down into metanephrine. One disease is going to use these breakdown products as a measure of catecholamine production. A pheochromocytoma is a tumor of the adrenal medulla chromaffin cells that produces high amounts of catecholamines into the bloodstream. You can use the lab values of these breakdown products to help diagnose a pheochromocytoma. This disease is going to be discussed more in detail later on in the organ systems. I just wanted to tie in a clinical correlation here. And since we're out of catecholamine coins, we're going to have to call game over for our catecholamine discussion. If you're following along in the first aid book, I would encourage you to take a look at the catecholamine synthesis tyrosine catabolism figure. I like this figure because it shows how some diseases are going to fit into this grand picture that we're painting. We should now go through some diseases that are caused by the dysfunction of amino acid metabolism. Phenylketonuria, or PKU, is one of the most common enzyme deficiencies, with an incidence of about 1 in 10,000 people. Since it's so common and important to detect, the USMLE loves to test on phenylketonuria. Make sure you know the mechanism of action and what amino acids are involved in this disease for test day. What causes PKU? Phenylketonuria can be caused by either an autosomal recessive deficiency in phenylalanine hydroxylase or by a deficiency of the cofactor tetrahydrobiopterin. Both of these are required to convert phenylalanine into tyrosine, so a deficiency in either one of these is going to build up phenylalanine and also lead to a tyrosine deficiency. This excess phenylalanine that we have can then be converted into phenylketones such as phenylacetate, phenylactate, and phenylpyruvate. These phenylketones get spilled into the urine, thus the clever name of phenylketonuria. Tyrosine is then going to become an essential amino acid. Since phenylalanine cannot get converted into tyrosine, and if it's not consumed in the diet, the catecholamines cannot be produced. So tyrosine becomes an essential amino acid required in the diet. Thinking back to our discussion on catecholamine synthesis, we've already seen this figure. Phenylalanine is converted into tyrosine, which can then be converted into our catecholamines. But also, without tyrosine, you cannot produce thyroxine or melanin. Consequently, patients with PKU will have intellectual disability, growth retardation due to the lack of thyroxine, seizures, fair skin due to a lack of melanin, they may have eczema, and a musty body odor, which is caused by this excess phenylketones. To remember this connection, keep in mind that the phenyl functional group is an aromatic ring, and patients will thus have this distinctive aroma. If you see the words musty body odor on test day, I want you to immediately think phenylketonuria. If a patient is diagnosed with PKU, how are you going to treat this disorder? Well, the treatment of PKU is to eat less phenylalanine and then eat more tyrosine because tyrosine becomes the essential amino acid. Since artificial sweeteners such as aspartame and NutraSweet contain phenylalanine, patients with PKU should avoid these. If you have a pop can in front of you, I encourage you to check out the label. Most diet pop cans will say something along the lines of phenylketonurics contains phenylalanine. PKU can arise in a fetus if a mother does not have sufficient dietary intake of aromatic amino acids. Infants from these mothers can have microcephaly, mental and growth retardation, and congenital heart defects. You'll see these same congenital defects if the mother has PKU and does not adhere to her dietary restrictions, since the phenylalanine and its phenylketones can then cross the placenta and cause damage to the newborn. Newborns often are screened for this disease 2 or 3 days after birth. The reason it's not immediately checked for is because some of the maternal phenylalanine hydroxylase enzyme may be in the fetal circulation during these first few days. Let's pretend a 3-day-old infant has elevated urine phenylacetate, phenylactate, and phenylpyruvate. What are the 2 possible defects in this 3-day-old? Well, this question is asking for what defects cause phenylketones to accumulate in the urine. The first defect could be due to a missing enzyme, phenylalanine hydroxylase enzyme. However, remember that this reaction is going to require a cofactor, and that cofactor is tetrahydrobiopterin. So if either of these 2 are missing, phenylalanine cannot be converted into tyrosine or its derivatives, the catecholamines or the thyroid hormones or melanin. Maple syrup urine disease is an autosomal recessive disease of amino acid metabolism, in which case both copies of the enzyme alpha-ketoacid dehydrogenase are deficient. This enzyme is going to catalyze the degradation of the branched chain amino acids. So what are the 3 amino acids that are branched? I want you to remember that isoleucine, leucine, and valine are going to be your branched amino acids. And you can remember this with the mnemonic, I love Vermont maple syrup, where I, L, and V are going to be your amino acids, and we're talking about maple syrup disease. Typically, these 3 amino acids are broken down into alpha-ketoacids, and then further broken down into acetyl-CoA, which can get fed into the TCA cycle eventually. This second step is going to occur with the help of our branched chain alpha-ketoacid dehydrogenase enzyme. Deficiency of this enzyme is going to lead to an increase in alpha-ketoacids in the blood, especially from leucine. Also, a deficiency of thiamine, or vitamin B1, will cause an increase in alpha keto acids as well, since vitamin B1 is used as a cofactor in this reaction. Why is branched chain amino acid disorder termed maple syrup urine disease? Well, affected infants are normal at birth, but eventually they're going to develop a characteristic maple syrup smell from their urine. So for step one, if you hear the buzzword maple urine odor or sweet syrup odor, immediately think maple syrup urine disease and the branched amino acids isoleucine, leucine, and valine. There are also CNS defects such as lethargy, feeding difficulties, coma, and seizures in maple syrup urine disease. If untreated, this can actually cause mental retardation or even death in the first month of life. So the treatment for maple syrup disease is going to be dietary restriction of your three branched amino acids. So don't eat these. And then you'll also want to supplement thiamine, which is going to be vitamin B1.